All right, so there's quite a bit in this topic. You'll see there are a couple slides that are repetitive. I pulled out some other ones, okay, tried to put in different examples and things. But mostly here I'm going to talk about the lettuces and a little bit about spinach and a few things at the end, okay, that are a little bit different. Okay, so, here what I like to do is start off with what are common considerations for this group. We know that water loss is an issue. However, it can work for us and against us, right? We often think water loss is bad, but not really, because a little bit of water loss makes the leafy greens more flexible in the processing lines. So I think that's something we haven't explored enough of. I mentioned that. Of course, mechanical damage, because that's associated with the microbial growth, a lot of it. Think of spinach, and I'll show you an example, right? Then here we talk about the nutrients. The darker green, sort of the rule is the darker green these products are, the more nutrients they have. Okay, so think you've got more chlorophyll, it's darker green, and directly associated with the chlorophyll is the amount of carotenoid. Both of those are in the chloroplast. The chloroplasts are mostly membranes made up of proteins. So the darker green, the more protein you have because of that chloroplast structure. So those are, necessarily more nutritively, more nutritional. And associated with that is also higher vitamin C content, okay? So if you want to go from one end, you think iceberg lettuce, right? Hardly any vitamin C, very pale, to something like kale. So that's sort of the spread. And all those things are related from a nutritional standpoint. Okay. And then they're sensitive to ethylene. Some are very sensitive, yellowing and a lot of the darker green leafy tissues. And then in some of the lettuces, we see more like brown spots and things like that, okay, rather than yellowing, okay? So those are most common. And all of these require low temperature. Anybody processing a leafy green that is chilling sensitive? Anybody? I don't think so, right? We have leafy greens that are chilling sensitive, but as far as I know, none of them are being processed as fresh cut, okay? So correct me if I'm wrong, if you occurs to one. This is a little bit different. I don't think you've seen this graph yet, but it illustrates, again, the importance of temperature and respiration. Here we have, and I'm going to use Fahrenheit, correct? Fahrenheit? All right. I hate using Fahrenheit, I'll have to tell you. It really goes against me. Okay. All right, 32 and 50, okay? But here we have then, they're not exactly the same, so we're comparing like oranges with mandarins here, okay, sort of, okay? So here, but we have immature leaves of a range, and then we have some mature heads, and these happen to be all lettuces. You can see the temperature. You see if you're at 50, there are differences in the groups. We know some are higher respiring, like the brassicas, than the lettuces. But once you move into the low temperatures, again, you get close to zero. Really, the respiration rates, there are differences statistically, but they're not important, right, okay? So again, the role of temperature there in minimizing then the metabolism of the products. Now, this was something, I'm going to put a big circle there. All right, this was something when we first started, we did a big study, actually, with a company quite a few years ago. But we were comparing lettuces, and these were the European salads at that time. That was the high-end European salads. And they were from 5 different companies, okay? Now, the company D, so you can look at the mixture there. The company D had more problems with shelf life. So you look at those products there, you can see we have frisee, we have escarole, we have romaine, we have iceberg, we have radicchio. And what do we have in D that would make it more problematic? We have, actually, I think that's tatsoi mizuna. I don't think that was arugula. It looks like arugula. It could be. Doesn't matter. Similar, okay? And what are those? Those are brassicas, and those are much more perishable. And to boot, it turns out that the company was sourcing those once a week. So in other words, the shelf life was determined by a couple flavor components there, color flavor components, and they lost shelf life, meaning bad odors, right? Okay, in the bag, okay? And so when we have special components like that, which ones have to be the freshest? Those, because those are the ones that are going to fail far before the lettuces fail, et cetera, because there we have modified atmosphere to prevent the browning. So one of the things here is that, pay attention to those ingredients and make sure that whatever ingredient is the most perishable, that ingredient is always the freshest, okay? And so, of course, it didn't take them long to learn that lesson, okay? But it is a very important lesson. And, at the beginning, it was something, we didn't really think that much about until we began to have lots of problems with shelf life. You already saw, so there, yeah, shelf life, most perishable product. You saw this, I won't belabor it. Just I left it in there by mistake, really. Okay. So for lettuces, when we think of general storage conditions, and this would be for escaroles, lettuces, that whole group really, okay? So as close to 0, but not freeze. Remember, freezing is just below. For iceberg, it's about 0.2. Romaine is about minus 0.3. So they are very close to 0, but they're not at 0. So for shelf life, typically we say the intact heads about 4 weeks at 32, about 3 weeks at 41. We can get longer. We have to work at it, you know that. But typically that would be high humidity, controlled atmosphere. Now, here's one of the interesting things, and was always a conundrum when I first started working with these. In the intact heads, we actually have damage by CO2, yeah? And the bottom line here is that the lettuce still, when we cut it up, it still responds the same. CO2 is really not beneficial because we're actually preventing the brown pigments, but we're still causing some of the metabolism. That's one of the reasons sometimes you open the salad bags and they'll turn they'll brown fairly quickly once they hit the air atmosphere. And that's because you've already had the metabolism going on, just that you haven't done that final polymerization of the pigments to make the brown pigments. So you open the bag, now you have air, poof, now they're going to turn brown. So many people will complain about that. They open the bag, and if they don't reestablish the atmosphere, close it right away so that the CO2 builds up again, they'll get that browning. And it's partly because we act as if they're different, but physiologically, really, they're similar. Just that the cut-edge browning becomes so much more important than the damage caused by the CO2. And I'll show you an example in a moment, okay? They're ethylene sensitive, and again, there they vary a lot, okay, on their sensitivity, okay? And then for fresh cut processing, one of the important things, we did a little bit of work, but not enough really critical work. But in our work, we would say that if you're going to store the romaine and then process it, about a week, because the romaine, for example, has more phenolic compounds. As you store the product, you get more phenolics built up. That means when you process it, it's going to brown more rapidly. I had a graph I used to throw up, and it was always confusing for people, so I didn't put it, okay? But that's what it showed. So about a week for romaine, and here we're talking at storage of 3 to 5 degrees, 38 to 41 Fahrenheit. And then for the iceberg, less than 2 weeks. For the same reason. You cut the lettuce after 2 weeks and you're going to have more problems. Would you agree with that, those of you who work with lettuces? Yeah? Okay. So those guidelines, do you adhere to those guidelines, or do you have others that are stricter? Yes. Yeah, anybody. Yeah. So in other words, your intact product, it would be less than a week old, right? And your iceberg less than 2 weeks? Yeah? Is it? Okay, that's good. I mean, that's really good. But that we would say would be probably the limits at which you would want to store those products before cutting them, okay? All right? Okay, and please, you know, in these talks, you have something to add, please. Yes? Mm-hmm.
The same concept will apply to brassicas, broccoli, cauliflower?
Certainly, because remember, the longer we store the intact product, changes are occurring. The product is headed to what? To die, right? Okay? Yeah, and so, you know, the sooner we process it, when it's stronger and healthier, the better quality the fresh cut product's going to be. That's a general phenomenon, right? Okay, so, okay. Just the critical thing there, I think, is to know what is the critical period. And that's what we don't know about a lot of products. You know, storing them different periods, processing them, and keeping track of, okay, how did our cut product perform? And that's something you can do in-house, right? Yeah, if you have a doubt about a particular product, okay? Yes? Mm-hmm.
You said like optimal temperature is about 32 degrees.
Yes, as close to 32 as possible. Yeah.
Freezing is 31.6, so there's only a
Yeah,
4.
yeah, that's what I'm saying. It's very close, right? Yeah, so in practice, you're not going to be at 32, right? But the idea is you want to get as close to the freezing point as possible, because we know there, with every half a degree Fahrenheit or every degree Fahrenheit, that will lengthen, it will maintain the quality and lengthen then the storage life. So really in the past we'd say, no, we don't want to be anywhere near the 32. But as our former engineering colleague said, you know what, we should freeze more loads of lettuce, you know, meaning we should get closer there rather than have them have problems with decay, right, and other issues, right? So in other words, always that's a challenge. We want to get as close as possible. And with better equipment, we have less fluctuations in the equipment, transport equipment, storage room equipment, and so we can have tighter specs there, and we can get closer as the equipment has improved, right, and become have less swings and fluctuations in temperature, right? But it's good to keep in mind, you know, even in the lab, when we say 0 degrees Celsius, typically we have it set at 0.25 to 0.5 because also we risk freezing, right? Yeah. And so, but we'll say 0. But typically, sometimes on the slides now, you'll see it'll say 0.3 or 0.5 because it wasn't set at 0, right? Okay, all right, Yeah? Everyone clear about that concept? Yeah? All right? All right. So here are some, whoops, sorry. Let's go back here to look at some of the examples. Now, so here's freeze damage, okay? So frozen leaf, and I have these arrows. And so in the intact lettuce, during the desert production, the winter, if we have freezing in the field, what happens? The epidermis, the surface area, will separate on this stem. So this separates, okay, yeah? And so what happens is now bacteria can get in there and you get a lot of rot and decay, right? So neither do you want to send it to fresh market, nor do you want to process that, right? So the inspectors look for that kind of freeze damage. Now, if the heads are over mature, you can get what we call heart injury. And basically there, you've got this new tissue growing, but the head is so dense that it actually goes anaerobic, right? And it'll turn brown, right? So it's kind of amazing. When I first heard that, I thought, well, that doesn't make sense. I mean, the lettuce is here, you've got air. But no, the tissue is forcing itself up in there. It's so compact, the oxygen can't get at the growing tissue that is respiring very fast. And so you'll get this kind of discoloration, and that's obviously over mature. If the lettuces are old, do you ever see this where you get pinking on the ribs exterior? Wow, you should not. Okay, so what does that mean? That means the lettuce was stressed in the field sometimes, okay? That can happen. But more likely the lettuce was on the over mature side, and now you store it for a period of time. So we can have lettuce that's mature, we store it for a month, and you'll get pinking on the outer ribs. So it's age phenomenon. Age on the plant, it's over mature, and or age in storage that will begin to see the pinking, and that's due to what? Phenolic compounds, right? Because they're going to increase during the storage time. So when you see lettuce like that, even though you remove the outer leaves, you know that all that head now has a higher level of phenolics, yeah? So I would say typically for the fresh cut, it's going to have to do with maturity, probably, okay? So if you see a lot of that, I would say you want to look at the maturity because you're not doing yourselves any favor in terms of browning and things, okay? All right. Now, the top. There are some photos showing very ugly brown stain, we call it, when the lettuce is exposed to high CO2, okay? But this is out of a salad bag. So when you start looking at the iceberg lettuce pieces, you'll see they have kind of this kind of translucent, kind of a crystalline look. Have you noticed that? And that's due to the high CO2. So we're getting the same problem because of the CO2, just that those atmospheres control the cut edge browning and everyone's focused on that. So this then becomes a minor defect. But as I mentioned before, the physiology is really the same, just we're kind of playing tricks with the lettuce, yeah? We're controlling the defect that is the principal defect, and that is the cut edge browning. But the CO2 actually still can be damaging to the surface. And you'll get that kind of, you'll get a crystalline look first, and then you'll kind of get that brown area. Not that common. Yeah, we have to look. You know, we looked at hundreds of bags, and you know, we see that once in a while, okay? Yeah, is that clear? All right. So now, another one would be ethylene, okay? So this is what ethylene damage looks like on russet spotting, called russet spotting. Ethylene damage on iceberg, you get those kind of spots. In the other lettuces, it's not so clear. You get kind of blotches of brown. It's harder to diagnose, but a similar thing, kind of a brown discoloration, usually on the midrib of the lettuce. You can see here it's moved into the leaves because that's a very high concentration of ethylene, but typically you'll see on the midribs, and here it's kind of a few here and a few on the leaf, okay? All right? And of course, that's something that 1-MCP would block. So if the lettuce were treated with 1-MCP and we give it ethylene, we get no russet spotting, right? But it has to do with cost and value and all that, okay? So we know it works, but we don't use it, no? All right. Now, lettuces vary in composition. So this is just some data between a romaine, iceberg. I don't know the cultivars in this case. But here we're looking, you have more chlorophyll, you know, more carotenoids because of the chlorophyll. Sugars are about the same, more vitamin C, and even that's low in the romaine. The romaine vitamin C can be double that easily, okay? And that's pretty high for iceberg, as a matter of fact, okay? And then the phenolics. Notice here we got about double the amount of phenolics in the romaine as we do the iceberg, okay? And so, of course, we look at different varieties. They would be different, but that's pretty typical variation. I showed this the other day where you're going to get a buildup of phenolics as the leaves become older, right? We store them. The more mature leaves have more phenolics. We're going to get more browning, okay? Yeah? Is that clear on that one? Yeah? All right. All right. And then these are a couple rating scales. This is for iceberg. Again, here we're scoring the browning 1 through 5. That would be the rating scale we used. And then this was one for romaine, okay? So you know that we would use, we'd have those printed out, we use them as kind of guides, okay? Now we can reference those objectively. And I showed this photo about the different cultivars and the maturity. You already saw that. I'm going to skip that. However, here is a little bit of data, and these are the days. This was a big trial with a seed company. We had 6 cultivars, 2 icebergs, 2 romaines, and then we had 2 crosses between the 2 parents, okay? So it's kind of interesting. And the other interesting thing is that we had 3 trials of the 6 cultivars planted all in the San Juan Bautista experimental field, same area, same production techniques, except one we harvested in June, so the planting was a couple months before, right? June, one in August, and one in October. We expected to see big differences, but actually they were quite consistent. So that, you know, I was wondering, right? Okay, so because in the past, in variety trials, we perceived that in June the product was healthier, had longer shelf life than the product harvested in August or October, no? But in this case, they were very similar. So I've just averaged them across the 3 trials, and these are the days to discoloration. And in the photo before, notice that we said, in this photo, romaine 2, you already said, wow, that one browns more, right? Okay? And so the data, the actual numbers show that. If you look at romaine 2, you see we had less shelf life, significantly less shelf life in that romaine 2 than the other. And then I mean, these are days for it to discolor, right? Okay? So in other words, fewer days for it to discolor in the romaine 2 than the romaine 1. And then you can see that here, in that cross, and this is a puzzle, right? Okay, in the cross they actually discolored sooner, right? So the shelf life of the cross, which I remember with the people of the seed company, they wouldn't believe it, right? Meaning they were very upset, and I said, you know, I'm not here to make you happy. I'm here to, you know, present the results, right? Okay, and these are the results. But they were very unhappy because these crosses, they were already, you know, promoting them to the industry. But it turned out, for some reason, we don't know, they actually browned faster. What was the reason? I don't know. Maybe they know by now, right? Okay. But that was kind of interesting. And then you can see here the differences between the maturities in terms of the days to brown. So that's pretty, I would say, typical, right? But that's the most complete set of data I have in terms of cultivars across trials and things like that. Okay, so here's some more on composition. But here we're looking at heart leaf, young leaf, mid-size, and full size. And if you're interested in that kind of data, again, that gives you a little bit of data of the variation that can occur in the leaves across a head of lettuce. That's also a bit interesting, okay? But we have to deal with that, okay? So coming back now to the atmospheres. So remember that the CO2 above 3% is damaging, okay? But in the fresh cut, again, the edge browning is the major defect. And so the high CO2 then helps us control the discoloration. I'm sorry, that one's a little bit out of order. I meant to put it sooner, okay? So how does atmosphere help us? So here's a trial, and this came out of that trial I was just telling you about. So here we have the romaine 1, romaine 2 in air, and here we use the CA, okay? Now, some people, you know, they talk about, oh, you use CA, controlled atmosphere. No, we use modified atmosphere. So why do I use a CA? Because, as you know, in the modified atmosphere packages, it's not going to remain the same, right? So we do the CA. One is we're equipped to do that in a laboratory, okay? We don't have form, fill, seal, packaging equipment. But more importantly, when we collect data like that, those are reference points for the modified atmosphere packaging. Because here we have a specific atmosphere, right? We can change the atmosphere. And of course, it's not going to fluctuate with time, so it's not real world, but it is referencing. It's a reference point for real world packaging. Everyone agree with that? Some people think that they dismiss this because it's controlled atmosphere. But the point there is to use it as a reference. So what do you think about the level of O2 and CO2 that we chose for that experiment? What do you think? Is that typical of your packages or not? Yes, no? Who works with lettuces? Nobody's going to raise their hand now. Did I scare you? Or I'm sorry. I'm speaking too loud, too aggressively. She looks, yeah, that's what it is. Well, I can tell you that oftentimes the O2 is lower, okay, 0.4, 0.5, and the CO2 can be higher. But that's, not bad, okay? I mean, that's in the range, okay? So you'll see as we do experiments, we do modify that a little bit depending upon the company we worked with and what they consider. No, we don't want 0.9, we don't want 0.5, we want this. Okay, all right, that's fine, okay? But obviously we have a beneficial effect, okay? And we can maximize, meaning we can increase the effect of retarding the browning. But as we know, that if we drop the oxygen and raise the CO2, we have other consequences, right? So we always try to look for the least extreme atmosphere that works, okay? So that worked pretty well. Now here's some other data just showing the difference in visual quality, and here we have different atmospheres with CO2. So you see that here for the visual quality, we have to have at least 6. If we have 3% CO2, it's too close to air, even though we have low oxygen. So we have to have somewhere between 6 and 9% CO2 to gain the benefit of the atmosphere. We know that, okay? Yeah? All right. And here's, okay, but as we increase, so here's the same experiments, okay, 3, 6, and 9. And what this is showing is some of the fermentative volatiles. So if you're interested in that, you can see that, yes, the higher atmosphere with 9% CO2, because we're at, here in this case, low oxygen, 0.2%, we're going to get more of the ethanol and more of the acetaldehyde, okay? So through the years, we've collected some of that data. Why? Because that's going to contribute to bag salad odor, right? Okay, so that's why we make those measurements, okay? All right. Now, not all lettuces respond the same, so it turns out that our romaine and iceberg really are benefited by CA. So here we have, you know, overall visual quality, and the bright green and the blue then are the iceberg and the romaine, and it turns out the other lettuces aren't benefited quite as much, okay, by the CA. But since most of the industry, romaine and iceberg is, you know, 90% maybe, right, okay? And so that's, you know, our guidelines for those. But at least when we run experiments like that, we know that, yeah, the benefit's not as great for the other lettuces. And many times those other lettuces are not being processed in the same way, right? We already went through this, okay? So texture's tricky. You know, we do it sensory. You know, we had a scale. You snap it, is it crisp? And I mean, other colleagues saw that and they said, oh my gosh, I mean, you know, no, we use a texture analyzer. Okay, you'd have to do like a gazillion measurements in order to get data because of the variation, right, okay? But it is possible. You can do a cutting, a shear force, you can do penetration, but it takes a lot of time to do that, right? So texture turns out not to be so easy to measure. The other thing is, where do you measure it, right? So typically we would measure it in the midrib. That's why we would kind of snap the piece. Is it crispy? Does it snap? Does it not? So texture is not not so straightforward to do. It would seem it is, but it's not, no? And we've already talked about the aroma and the flavor in terms of those bag odors, and a little bit about the nutritional value, which is the vitamin C, and I have a different example to show you today. You saw this slide already. I'm going to pass that by, but I have to put it in there just to emphasize the importance of temperature and the relationship with off odors and things like that, okay? So here's some other data that you hadn't seen, and this came out of one of our recent publications, again showing that in most of the experiments, we would evaluate day 3, 6, 9, something like that, okay? And so I don't think I have an experiment we did like 2, 4, 6 days, okay? So somewhere between day 3 and day 6, in the controlled atmosphere, we lost a lot of the vitamin C. Now you can see in air, it's also declining, right? When we hold the tissue in air. That's a pretty big decline. Usually it's actually a little bit more stable, but it depends on the lettuce. So we know that the lettuce itself, depending on the health of the lettuce, it will retain the vitamin C, or the other trend is it will be a fairly steep decline. And in that case, we had a fairly steep decline. But the atmosphere really further decreases the vitamin C, yeah, okay? So it's something interesting, I think, that, you know, that should be explored if we're thinking about the lettuce as a good contributor of vitamin C, which the dark green leafy lettuces are. But obviously, those atmospheres can be somewhat problematic, and so there are some alternatives. Here, clean washed leaves, but they're not in modified atmospheres, just air packaging, right? We have the romaine hearts, yeah. Then we have other specialty leaves that are mixed washed. Again, no modified atmosphere packaging. And in this case, the ends, when they're removed from the head, they will brown, right? But all a chef has to do then is one whack, and usually you don't cut your fingers with just, you know, one cut, right? And so that turns out to be a very good product. Very immature lettuce heads, etc. And then one company that used to be in the fresh cut business, that's TNA. They used to have a fresh cut plant called I forget now. It's been a while. Eh? Fresh Thyme? Fresh Thyme. Okay, thank you. Oh, Salad Thyme. Salad Thyme. That's what it is. Okay, yeah. That didn't sound quite right. Okay, Salad Thyme Farms. But then they got out of it, and they've taken a different strategy where now they have the different lettuces so that you can make your own salad in different ways. So in other words, there are different strategies to get at the same kind of diversity that some of the salads and the color and flavor components can add, right? Okay, so different strategies in the industry, which is interesting. We were involved in a project about romaine hearts because starting off with this is one of the first photos I was shown. So you see that photo there, you got a thermometer sticking out there. Okay, anybody have a thermometer with them today? Okay, where's your thermometer? Okay, all right. By the way, it's also, I like it because, you know, okay, yeah, so I tend to carry it around when I'm in sketchy areas, okay, so shall I take your temperature, you know? Okay, anyway, in that photo, which was from a shipment, what we see is 41 Fahrenheit. What do you think about the temperature? This was destination, arrival. It had been, a little high, don't you think? Yeah. And what do you think here about this? You see you have the pinking on that romaine heart, and what's that due to? What is it? Immature? No. Damage. So, cracking. Notice, you have a crack there, right? So you have a wound. It's like if you cut it for fresh cut, what is it going to do? Brown? Because in that package you've got holes, right? No modified atmosphere. So the industry very kindly gave me some money for a project and said, solve this problem, which of course is not easy to solve, right? But the main thing we came up with were two ideas. One is, obviously the 41 is too high. That's sort of like a no-brainer, right? Okay, so the first thing to do is you go with low temperatures, 0.5 Celsius, 33, okay? 2.5 is 36, and then the 41, okay? So we did a series of experiments, but this is probably the most illustrative. And then over here you have hours, 1, 3, 6, 9, 12, 16. What is that? Delays to cool. So within the operation, the idea was, what can they do without putting money or that would cost them more? And one is just being more efficient. More efficient at temperature management, right? They cool it down to low temperature, but then by the time it gets shipped out, it's a little warmer. On transportation, a little warmer. So paying more attention to the temperature, but also paying attention to the delay to cool. So sometimes it was very fast, you know, field's very close by. Other times it was warm and the field was far away. So here you can see at 0.5 doesn't make too much difference. 16-hour delay, you get a difference in shelf life over 21 days. So that was the thing. We want the lettuce to last 21 days and be marketable. But at 36, you begin to see now the delays to cool make a difference. And again, up here, now we're at such a high temperature, the delays to cool are not so important because now that temperature is high enough to allow the phenolic metabolism, even if we cooled the lettuce rapidly, okay? So right there, that intermediate right there is the interesting one. So 36 is very reasonable and paying attention to the delay to cool, okay? So that was the most important sort of information. Okay, then we have different romaine varieties, and they differ in their browning. And I had, I showed broccoli actually. But you know, we did some variety trials, and like I say, it's like an abyss, right? There's so many varieties to evaluate, no? And I hate doing that, but you know, that's something the industry is always interested in getting feedback on the varieties, okay? So you can see that kind of variation, which again can help us or disfavor, right, our ability there. So moving on, water jet cutting. Who's familiar with water jet? Anybody use it in their plants? Yes? You use it for?
I haven't seen it yet.
Oh, okay. Okay. Okay, very good. Okay. Okay. So my understanding is right now, I think it's Ramsey Highlander uses water jet in cutting romaine in the field, and then it's used here for cutting celery. And Jeff, do you know if it's used in any other products? Celery, right.
Is the big one.
Right, exactly. Okay, very good. And that's what my understanding too. I'll show you an example in a moment there. So anyway, we did a project, which again, talking about an abyss of use of time and things, it was really a big use of time here. We had these different orifices, we have different pressures, and then we had these speeds at which you cut. So the company thought that all of those things made a difference. That was a bunch of baloney. The only thing that makes a difference is the orifice, whether it is a sharp stream or a standard stream. So that was the key. The rest of it didn't make any difference, really. I mean, there were slight differences that weren't important. So it had all to do with the quality of the water stream. And I showed you then this photo before, okay? So here was our best example. This is a food grade nozzle with a very fine orifice. And here this lettuce, this is romaine, cut romaine. It's been stored 18 days at 36, no modified atmosphere. So why are we interested in doing this? Because we're interested in trying to find different ways to reduce the browning susceptibility of the lettuce so we don't have to use such extreme atmospheres. Because we know the atmospheres are bad for texture, bad for aroma, bad for vitamin C. Yeah? But we haven't solved it yet, but that's the idea behind it. Anything we can do in the cutting, the varieties, all of that that contributes to having less browning, okay? Is that clear? Yeah? So anyway, we have...
I can tell you that our laboratory is doing tons of testing for seed companies
Yes.
on this very topic.
Yes, very good.
Constantly coming up with, through conventional or GMO or whatever
Yep.
way,
Right.
ways
Right.
that they can get iceberg and romaine not
Yes.
to
Brown. Right.
in an oxygen-rich atmosphere.
That's right, because that allows us, again, to have lettuce that has a more normal characteristics.
Very seriously.
Yep, very good. I'm glad to know that. It's been a long time coming, but very good. And it is a process, right, all of this. So here's some data, and I won't belabor that, but here we're looking at visual quality, discoloration, and then whitening, okay? So there are two graphs there. This one has to do with the different orifices, and then this one had to do with, and then this one had to do with two pressures at the best nozzle, and you can see, you know, they virtually lay on top of each other, right? Oh, sorry, let's see. The food, the open are the food grade, and then the black are the knives. Okay, yeah. So you can look at that data if you're interested. And we did a little microbial work, and you know, initially the water jet, the microbial load's a little bit better, but after it's stored for a period of time, it's very similar. So you don't gain an advantage there. So here we created this white scale index because it is true with the water jet cutting, it's an erosion, and you'll get more whitening on the cut edges. So we noticed that. So we had this scale for the lettuces, but that's much less of a defect than, say, browning on the edges, right? Okay, so we can tolerate that. All right. And so, okay, that's our post-harvest evaluation. I don't think there's anything there. I'm missing one. And what I was missing, oh, no, we'll go on. Okay, I'll show you an example of celery and the white ends of the celery with water jet, okay, in a moment. Okay, so now I have like 10 minutes or so. Let me, all right, I'm going to talk about spinach, okay? So some quality characteristics. One of the interesting things here, in many of our leafy greens, we over-fertilize, so we have high nitrates, which they pay attention to in Europe, but we tend not to. At the end of the presentation, there are some references about nitrates, okay? Yeah, so it's been something you're kind of wondering about, but we tend to over-fertilize with nitrogen fertilizer. Spinach can accumulate oxalates, but the good news is that the baby leaf spinaches, which dominate the market now, they're young leaves, and therefore they tend to have low oxalate contents. It's the big mature leaves that will accumulate a fair amount of oxalate, and that's kind of bad for us for kidney stones and things like that. So for storage, it's very similar. High CO2 atmospheres are also stressful for spinach, yeah, it turns out, yeah. And you can see our storage time is a little bit less than in the lettuces, okay, more perishable. So for me, one of the big things about spinach is what? You know, we would go out to the field and we would bend over, we would have a scissor and we would clip the spinach, put it in a little bag, you know, and we get back to the lab and half of it still had cracks and breakage. You know, it was driving me crazy. We couldn't do an experiment with, you know, number one, I mean, you know, it's hard to get leaves that don't have damage, right? So damage, right? And damage leads to what? Bacterial growth, leads to off odors. So it's really a big problem, okay, the damage. So initially we used a scale of 1 to 5, and basically we streamlined that and we just said 1 to 3. So we basically said, okay, 1 and 2 go together because you hardly ever have category 1. And then you have category 3, moderate damage, you can see it. And then you have small pieces and others, which is really a problem, right, okay? So those are the ones we really worry about. So in the spinach, what I want to talk about is this concept about ammonia and the leafy greens. And this is something that was supported by the California Leafy Green Board for a year, and sort of proof of concept. And the idea here is trying to use the analysis of ammonia as a freshness indicator, okay? Yeah? And so the idea is that when you first harvest the product, it's very healthy. But as it ages, you have a turnover of all your constituents. And part of that's your amino acids and your proteins. And part of that turnover is you produce some ammonia. And if the tissue is healthy, the ammonia is toxic to the cells. Have you ever seen ammonia leaks in the cold rooms, right? Okay, what happens? Things turn black, right? Okay, ammonia is very toxic, right? But it's toxic inside the cell too, right? So that ammonia that is released has to be reincorporated. Otherwise, it's very damaging to the cell. It changes the pH and screws up the metabolism, okay? And so it has to be reincorporated. There's a particular enzyme glutamate synthetase, okay? That reincorporates the ammonia. But when the tissue ages, that enzyme's less active, and I don't think I put an example in there, okay? And so what happens? Ammonia begins to accumulate. So for me, it's very interesting for the leafy greens. The initial levels can vary a little bit, but they're always low. And always during post-harvest, the ammonia increases. It increases if we give it ethylene. It increases if we increase temperature. It increases if we give the product stressful atmosphere. In other words, it responds to a lot of the post-harvest parameters that we try to manipulate to maintain shelf life, right? Okay, so here, ethylene, atmospheres, temperature, and time. So even if we have at low temperature, with time, the ammonia will increase as the tissue ages, okay? So for me, it's like, wow, I mean, this could be potentially useful for us in predicting quality and shelf life. So now going back to the spinach, let me show you one graph. So this is odors, whoops, sorry. So this is odors, off-odor score, and of course, we had more decay and stuff, okay? And this was ammonia. So we show that the ammonia is related with the breakage, is related with the decay, okay? Yeah, so now going back to this graph. So here we have 3 levels of damage. So we have slight damage, moderate damage, severe damage, and these are days at, two temperatures, 36 and 45 Fahrenheit, okay? So we have decay on the left, and then we have ammonia on the right, okay? And you can see how higher temperature results in more decay, more ammonia. Lower temperature, we have some decay, and we have less ammonia, right? Okay, so we did a lot of experiments like that. What is the association in time of the increase in ammonia with different parameters? So we studied that through quite a few years on different leafy greens, okay? So here's one experiment. That's, of course, a beautiful experiment. That's why I'm just going to show it to you, okay? But what's interesting here is that usually the increase in ammonia was measurable when already we could see a change. But if we could measure the ammonia and not look at the product, okay, we know they're tightly related. In this experiment, which is what I was hoping for, the ammonia actually increased before we were able to detect decay or off odors, yeah? So that's what I was hoping for, that it would precede, some measurable increase in ammonia would precede the other attributes that we could evaluate, okay? And this is one experiment where it did. So here we have 3 temperatures. We have our visual quality yellowing score. We have an objective color value. We have ammonia, and we have, yeah, and so that's in the, that's time course, right, for spinach. So that's actually very nice data. I'm very proud of that data. Okay, now, so what is the problem, though? The problem is, how do we measure ammonia? We take the tissue, we grind it up in water, we have a colorimetric assay, we need a spectrophotometer. It is not so simple or straightforward. It's not something you can do on a rapid basis, right, okay? So when we do the experiments, what do? We take the tissue, we put it in the freezer, and then we run a bunch of them at one time. So the idea here for proof of concept would be, okay, you're producing ammonia. Ammonia it's going to be soluble in the cell, sap in the cell, but it's also going to be in the atmosphere. So can we detect it in the atmosphere? And of course, there are ammonia detectors for leakages, right? But we're talking about orders of magnitude difference, right? Okay. So when I first talked to the industry about this project, they said, yeah, well, there are ammonia detectors. Go buy yourself an ammonia detector. I said, no. I mean, we're talking about, tiny amounts of ammonia, right? And so it turned out that finally I found a person at the Georgia Tech Institute, by the way. I was an engineer, and she was working with ammonia sensors for bomb-making materials. So this would be like people putting bombs in FedEx and UPS and all those good things, right, okay? So she was working on a project there, and just by chance, sort of you talk to somebody, you mention something, and, I found this person, okay? So she had some sensors, and so the support from the industry really was proof of concept. So here, I'm going to show you some data. So we did a couple experiments, okay, but this is one I think that's probably more interesting. So here we have spinach, commercial spinach. It's 6 days at 32, 6 days at 50, 2.5 days at 68, and 6 days at 68. So we had 4 situations, okay? So then we measure the ammonia. That's the ammonia, okay? And then she measured the ammonia. Now it was tricky. I took the tissue. I had to fly to Georgia Tech. Then what we do is take the tissue and put it in a bag because it's a volatile, right? So we have to trap it somehow. So we put the tissue in a bag. We waited like a few minutes, 30 minutes maybe. We had a standard. Does it say? It doesn't say. Anyway, and then we measure the ammonia with the sensor, okay? So proof of concept, you can see that, okay, this one and this one look quite similar. So 6 days at 50 Fahrenheit and 2.5 days at 68 gave us a similar visual result, okay, similar. And you can see the sensor also told us it was similar, okay, yeah? And then this was the worst one, and it had the highest ammonia, okay, yeah? So as proof of concept, you can see that we could measure the ammonia in the volatile phase, okay? Now, we didn't get any further funding because the industry said, how's that going to help us, right, okay? Somebody in transportation, somebody in handling, that might be useful for them. But as growers, shippers, we don't see the value of this. But as proof of concept, it should work. So I've been kind of out there hustling this idea, okay? You know, I'm retired now, you know, but I think this really could potentially be a useful freshness indicator. And that's really how we're proposing it for leafy greens, okay? Yeah? So just kind of putting that idea out there, okay? All right? And then there's some more, you know, information. We've done a couple papers, and there are other papers. I'm not the only crazy person talking about this. I mean, there are others who have published on this idea, right, okay? Ammonia is used for fish, freshness of fish, for example, okay? And there are other techniques, but again, the laborious techniques, what we want is a rapid response sensor. Oh, the other thing about the sensor, guess what? You completely destroy the sensor for like a whole day if you get moisture on it. So what do we have in our salad bags and things? Blah, you know. And so, but now I found an engineer here at Davis, and yes, there are other sensors, there are other ways to detect ammonia that would be possible, okay? So, yes, yep, 2 minutes, oh yeah, okay, all right. So, you know, I think still the idea has some validity, okay? So, you know, what's the application? Mostly leafy greens, that's how we're thinking, leafy greens, brassicas, that type of thing. Okay, and then the last thing, I showed you this slide before about the difference in dry weight, remember, of the trials, how important it is? And something, as I worked through the years on different lettuces and the baby lettuces and things like that, you recognize that we have some dehydration that occurs. It's not all bad, right? Because this product, for example, ended up being put in a plastic bag for a market that had no refrigeration. It worked better this way than if it were very fresh and crispy, okay? We have the breakage, and then you have, you know, you have different amounts of shrivel in the field. So I think there's a whole bunch of information that we don't know about water loss and how it can work for us in making the tissue slightly more flexible, but not having too much water loss that damages the product. And so we had little bits of information. We went into a plant, spinach, right, and we were, you know, weighing the spinach. They really thought we were crazy. But anyway, weighing small amounts of spinach and running it through the line after we had created a certain amount of water loss, right? So how did that spinach store? How did it? But that's the type of thing, is understanding what we call in general water relations. The water relations of the leafy greens and how that can help us in the salad, packaged salad area, right? So there's a lot, I think, to do there, okay? All right. And then the last thing, almost the last, I have like minus 1 minute, I know, is some colleagues have been talking about the use of light in the storages, right, in the retail market. If they're exposed to light, the lettuce could still photosynthesize. And so there is a couple publications, but notice the temperature. It's like, I almost shot him when I saw that. I said, why are you doing the experiment at 12 degrees Celsius, which is like 55? And he said, well, because, you know, at lower temperature, it didn't show a difference. I said, oh, wow, you know, okay, you know, so, but anyway, there might be something there. And then the last thing is, you know, there are a lot of changes in our production systems and things. And this comes back to the idea of the nitrates, and that is because in those hydroponic systems, you're able to have more control, right? You fertilize it, you grow it, but then you can redraw. Santa Maria, a researcher in Italy, they would take away then the nutrients a few days before harvest and drop the nitrate levels. So there are things to do there that also nitrates are considered anti-nutritional, right? Because they convert to nitrites, they can tie up the hemoglobin in young people, old people. And so there's that factor where now we have more control over the production conditions, and that will change the quality of the leafy greens, right? Okay, we can do that very, you know, specifically. And that's all I have, okay? Thank you.